radiology. Just as an intro, so the whole session is going to be recorded, but then we will have the ability to edit this, and after that it's going to be posted on YouTube. Milena has great experience in collaboration within European networks. She was participating in Horizon 2022 projects, and she's a she's a president of a Serbian Society for Cancer Research that is affiliated with European Association for Cancer Research. Today she's gonna give an introduction that refers to cancer medicine, but focusing on rectal cancer predictive biomarkers. So Milena, the floor is yours. Thank you. Now I will stop sharing my screen.
Perfect. Thank you very much, Polina. Thank you for the nice introduction. We are very happy to be here with you today. So yes, as you've stated nicely, we do actually have quite some experience with the Horizon Europe projects, Horizon 2020 and cost actions, as well as we've been participating in many of those in the past years. So please let me know if you see the presentation.
Not yet.
Not yet? Okay, let's see why.
I only see information has started screen sharing.
Okay.
Yes, it looks like that it's coming, but still on the way.
Yeah. Okay, well, so I can give you a short intro then about our group now. So let's resolve the technical issues if not. So the group that is presented here will be myself and my two PhD students who actually work mostly on this rectal cancer project that we are very proud of. It's the first project that we've received as a coordinating hospital institution from Serbia, which I think is really good for ironing out the inequities in cancer research which we very often encounter especially in Eastern European countries. So in 2022, we actually received this grant from the Horizon Europe funding scheme, and it's a twinning project that's actually a collaborating project with 3 higher institutions, and it is focused on rectal cancer. So for now, I think that I can say that it's progressing very well, and we are very happy that we can also share it within this cost action with you. So if the sharing has not started yet, I'm gonna stop it now. Let's try again. Is it okay now?
No, there's still just information that you have started screen sharing, but it's like not displaying the presentation. You don't see the presentation.
Okay, not sure why, because...
To try, do you want to try to open?
To try what, Mladen? I didn't hear you, sorry.
To open presentation.
The presentation is open, and in my screen, it says that it's sharing. That's why I'm not sure what's going on. So, not sure what's happening. Let's try again. How about now?
Yes, now we see that.
Okay, perfect. It was some kind of technical issue. Okay, great. So what I would like to focus on today is to pass a little bit through the project itself. But of course, as we are in a cost action, which is focusing on translational control, and we are very happy to be here with all the cost attendees, I will go briefly into the cancer medicine of rectal cancer. I'm not sure how many of our cost participants are actually in this kind of studies, but I think it's very important to get an overview of the world situation, because usually rectal cancer has been put into the same category as colon cancer in the colorectal cancer entity. But actually, if you want to be biologically really clear and clean, it's a very separate biological entity and clinically as well. So what we have actually seen rise in incidence, and of course you can see the numbers about the new cases and about the deaths that actually happened. These are data from 2020, and what's important to see is that higher income countries are not spared of these incidents and death tolls. So although there are better treatment options, the incidence is not declining, and the mortality is not declining either. So there are some very bad ideas that these kind of cases will be even higher up in the coming years. So what we've identified in Serbia as well, and it was an issue that arose from the clinic, from our clinicians, and we are very proud that we actually did, the real translational project derived from a clinical problem, which our physicians told us that they have, is to find a way to deal with this problem within our country, starting from our institution, to have a pilot project that will address this issue in rectal cancer, and then to use it as a proposal for changing the national guidelines in our country. So coming back to the rectal cancer itself, as you can see on the images, and Mladen is our attending radiotherapist physician. He will be continuing to talk after me, so he can explain much better the clinical aspects of rectal cancer. But although we all put it together as an entity in colorectal cancer, it's practically differently treated, and the biology behind it is different. So of course, the screening and diagnosis and everything else starts from a regular checkup. So we do have a national screening program for colorectal cancer in Serbia. I know that it is progressing as well as it can be in a country as ours, but it could be much, much better. And I think the accrual rates for our screening are not as high as we would like them to be. This can also be one discussion point for countries of other cost action participants, because I think a lot of countries do have a problem with the screening program in different countries.
Excuse me, Milana, I'm really sorry to interrupt you, but what I see is the introduction slide. Are you changing slides currently?
Yes, I am changing slides. Are they not changing?
I don't see that.
Okay, so not sure why. Let's try to share it again then. Do you see them now?
I only see the introduction.
Again.
But we see the it's like the
now we
PowerPoint.
see, yeah, that's the
Yeah, if
PowerPoint.
you change it there, it's good. Yeah.
Okay.
Yeah.
Okay, let's change it like this. Maybe the full screen issue is bringing some problems as well.
Okay.
So if we relate to the guidelines that have been published in 2024, so you can see what are the symptoms, and also this is very important for us to track concerning this cost action. If you want to talk about translational control, what are the biomarkers that are actually behind rectal cancer, which affect the treatment, and of course, by consequence, they do affect the way that we do science behind rectal cancer. So much of our projects are guided towards solving the issues in the clinic and what we can do actually to enhance the diagnostics on one side, and on the other side to advance the treatment options that can be applied throughout the world. So these are some 5-year survival rates, which of course are not very good, depending on the stage, of course, and on the treatment options that are present in each country. In Serbia, it's not so bad, but it's not as good as it can be. I think Mladen will shortly also talk about that later on. So what we also want to say is that the genetic aspects and the biology of rectal cancer behind the story are really strong. So colorectal cancer is guided also by some genetic changes, and we are trying to find each and every one of them. We want to act, of course, as early as possible. We want to stop this progression towards cancer. We want to catch it in the progression part from the normal epithelium towards the cancerous tissue. But of course, it's not as easy. And different parts of our projects are addressing different types of this translational control. So this specific project that we will present today focuses on treatment options for rectal cancer. But I just wanted to say that this is a very broad topic that we are working on at the Institute, and that we would, of course, within this cost action, be very happy to collaborate with other groups who are maybe focusing a little bit more on the control of progression towards cancer as well. So what's important in this field, I think it's very interesting to see what are the current guidelines for the testing, which, as I said, of course, simplify the situation a lot, but that also induces the treatment options in various countries. So by NCCN guidelines, you can see RAS and BRAF testing is, of course, obligatory. We can talk about different things in different countries, but in Serbia, we do the generalized RAS testing for our patients, depending, of course, on the National Health Fund reimbursement. Here, the guidelines are a little bit tighter, so there are stages of patients that we can test and those that we cannot. HER2 amplification is, of course, also recommended, and MMR and MSI status, of course, if it has not been done previously. So in Serbia, of course, we started testing this as well, and treatment options do include chemotherapy, surgery, radiotherapy, and immunotherapy. What NCCN also thinks is concerning these mutations that I think are important for this cost action is that testing should be conducted as a part of broad molecular profiling. So not sure how the situation is in each country, but in Serbia, this still does not happen. So we are at the level of single gene testing as well, which of course we all know is not very good for patient samples, and it's not very good for the turnaround time. So it would be nice if we could accompany the guidelines which say that both tissue and liquid biopsy should be tested, and that NGS, of course, is recommended. I hope that a lot of countries in the cost action are doing it. In Serbia, we do it by qPCR, so single gene testing. Of course, it's a multiplex PCR, so there are many RAS changes that are tested at the same time, but it's gene per gene, which is, of course, not very good. This is on one side the diagnostic setting, which I think is a reality for this story. And the other thing is the science behind it, which we are trying to progress, and we are working towards more European projects, which will enable us to progress towards this NGS-based testing, both from tissue and liquid biopsy, which I think is really important at this moment. So coming back to the project, which I mentioned at the beginning, it started in 2022, and we were very happy to try and solve this one small clinical issue within rectal cancer, which was derived from our clinicians saying that treatment of rectal cancer needs to be more advanced, that we need to find the specific cohorts of patients which are treated in the same way, but which do not respond the same way, which I think, of course, is the main task of our scientists, especially of those who work in a hospital setting, which do have very good access to patient samples and which do have very good access to clinicians, and we can collaborate with them closely. So I think this success story was derived from all these factors. So our mission was to find predictive biomarkers of a specific profile cohort, which is neoadjuvant treated locally advanced rectal cancer, stages 2 and 3. The chemoradiotherapy part will be better explained by Mladen, and he will touch upon some radiomics issues that we had within this project. But what we wanted to see is actually, is there a cohort of patients within this group that can be spared from surgery, applying the so-called watch and wait approach, which was currently not available in Serbia. So all patients, regardless of their clinical and pathological response after neoadjuvant treatment, progress towards surgery. And we wanted to see if on one side there are patients who can be spared, because we all know what surgery brings forth. And on the other side, if there are some really bad responders that actually can be switched to another treatment. So this project started last year, and I'm happy to say that we're collaborating with 3 very nice higher institutions, which are supposed to transfer also expertise and knowledge towards countries which are still in development, like Serbia. And we want to expand the cohorts of patients with many other countries as well. And this is also an invitation to other cost participants. If you have cohorts of patients which fit into this criteria, we would, of course, be very happy to collaborate with you on this project and on something that can be derived from that. A short representation of our institute, just so you have some idea what can be done and what kind of collaborations can be achieved. We are the National Cancer Research Center, so we do science, diagnostic testing, but preferentially treatment. There are around 1,000 employees. We have the clinical trial sectors as well. And we are proud to say that our hospital and research department work very well together. So we've been able to get some international funding as well in the past years. So today's story will focus on the evolution of this project. We started relatively small. We wanted to see if these clinical and molecular markers of response can actually be found. These were PhD theses of the two students that you will see after me today. And I'm very happy to say that they're both progressing nicely. Mladen is supposed to defend his thesis this year, and Alexandra will defend next year. So I think the project behind the whole story actually made them evolve both personally and as a team, which I think for this network of young investigators within the cost action is really important. I'm sure that you also work towards profiling these young researchers into the next generation of people who will actually lead all these stories coming forward. And I'm happy to say and to offer Mladen and Alexandra also for all collaborations independently of our group, because I think they're excellent young researchers which can contribute at the EU level very nicely. What I wanted also to say is that all these projects, I think, are nice because you start small. We started with a team of 5 people. But as the project developed, this is the network that we currently have from 4 participating institutions, and it's a perfect mix of clinicians and scientists, and also project managers, which I think is important. So coming back to this network of young researchers, I think these webinars are a good place where they can connect and also talk to each other afterwards, because we need these projects to progress. Coming back to the science of our project, this was the scientific workflow. Of course, 10 minutes is not enough to tell you everything about what we wanted to do. For this cost action, perhaps the genomics part would be the most interesting one, but we still don't have enough data on that because it's in progress and we could not share it today. So we started with some data mining and in silico analysis. We progressed towards proteomics, genomics, and radiomics. Some studies are finished. Genomics has been started. Metabolomics has not yet been initiated, but it's en route. And what we actually want to achieve by September 2025 is to integrate all these omics data that we should have by September next year, and to find this translational applicability. What we want to see is actually a pipeline of molecular analysis that can be applied in the clinic and say, okay, these are the patients that could be spared of the unnecessary surgery, progress to a watch and wait approach and a strict follow-up by our clinicians, enhance their quality of life, and of course, save on some funds for treatment as well, which in Serbia is a big factor. So we've been very happy to publish already 4 experimental papers from this project. We will be focusing on these 2 today. Alexandra and Mladen will present some data that we had from radiomics and proteomic studies. I think they are quite important because in rectal cancer, the science has not progressed so much. As I said in the beginning, everything was focused on a mix of colon rectal cancers. So it's very difficult to find a very nicely profiled rectal cancer cohort. We are very happy that we are advancing the science behind this field. And of course, I would like to invite you all to check our publications and also all the things that can be found on our website, because we are working towards many other things as well. Science is, of course, the focus, but advancing the science in countries with limited resources is also something we are very passionate about here. So I think that's fine for the introduction for now. We should maybe switch to the presentations of the young researchers. This overview, I think, guided you nicely into what we wanted to show. So now I think Mladen will start with his presentation first, and we'll be here for the discussion. Thank you.
Thank you so much, Milena. Great. So, Mladen, the floor is yours.
Thank you. Okay. You can hear me, and you can see the presentation. Okay, great. Hello from my side also. I'm Mladen Marinkovic. I'm radiation oncologist at the Institute for Oncology and Radiology of Serbia, and teaching assistant to the Department of Clinical Oncology with Radiotherapy at the Faculty of Medicine, University of Belgrade. Today, I will talk about pre-treatment MRI or radiomics in rectal cancer chemoradiotherapy response prediction. For the beginning, locally advanced rectal cancer is the rectum is the final part of the digestive system between the sigmoid colon and anal canal. It is located 12 to 15 centimeters from anal verge, and locally advanced rectal cancer is defined as T3 to T4 and 0, or NET stage N+. It is second and third stadium of disease. The best way to define rectal cancer stage initially is combination of pelvic MRI and proctoscopy examination. Here we can see example of the rectal tumor located at 7 centimeters from anal verge on MRI sagittal plane. Also, before treatment start, it is important to exclude the distant metastases with CT or chest radiographs and either MRI or CT imaging of the abdomen. And locally advanced rectal cancer is indication for neoadjuvant chemoradiotherapy followed by surgery. After completion of neoadjuvant treatment, response assessment is done 6 to 8 weeks after treatment completion, with MRI and endoscopy examination. According to that, clinical complete response can be found. It is defined as the absence of clinical endoscopic or imaging evidence of the disease. Here we can see example of clinical complete response after neoadjuvant treatment assessed with MRI and endoscopy. Here we can see the white scar in the place on the initially located tumor. For patients with initially distantly located tumor and clinical complete response achieved, it is optional to omit surgery, follow-up patients, and only in the case of local tumor regrowth to perform surgery. It is beneficial for quality of life of these patients because for distant located rectal tumor, the surgical strategy is abdominoperineal resection, and it is associated with permanent colostomy, so that influence on the quality of life of patients. On the other hand, for patients where surgery is conducted after completion of neoadjuvant treatment, response assessment is done using tumor regression grading system from TRG1 to TRG5, where TRG1 means pathological complete response, no evidence of disease in pathological specimen, and TRG5 means that it is there is no response on the neoadjuvant treatment. Nowadays, in the locally advanced rectal cancer, we have a lot of treatment modifications. Treatment adoptions are going towards prolongation of the period between completion of neoadjuvant treatment and surgery, changing the type and regimen of chemotherapy with intensification of neoadjuvant chemotherapy. It is called total neoadjuvant approach. and also their radiotherapy doses can be increased. According to that, there is a strong need for predictive biomarkers in locally advanced rectal cancer settings to personalize treatment of patients with this disease. And predictive biomarkers will enable the better local control and survivalness of these patients. And also, it can help us to select patients for watch and wait approach where surgery can be omitted. In our study, we collected 75 patients with locally advanced rectal cancer. All patients were treated with long-course chemoradiotherapy. Radiotherapy was conducted using volumetric modulated R therapy, simultaneous integrated boost. Here we can see the image of radiotherapy dose distribution. The red area represents the primary tumor region, where the higher dose per fraction was applied. It was 2.16 gray per fraction, and after 25 fractions, the total dose was 54 gray, and the blue-green area represents the potentially microscopic disease presence region, and their lower dose per fraction was applied. Also, concomitant chemotherapy was given during first and fifth week of radiotherapy, and it included a combination of fluorouracil and leucovorin. Response assessment was done in the 8th week after chemoradiotherapy completion with control MRI scan and rigid prostoscopy. And according to the response, we divided patients into responders and non-responders group. Responders were patients with clinical complete response. These are patients who were enrolled in watch and wait program without surgery, and TRG1 and TRG2 postoperative categories for patients who were operated. TRG1, one more time, means pathological complete response, and TRG2 near complete response. On the other hand, non-responders were patients classified as TRG3 to 5, according to pathological report. And at the end, responders comprised 35 patients, and non-responders group included 40 patients. I said that all patients had initial MRI to define rectal tumor stage. Here we can see information which we can retrieve from routine radiological report. Some of them are established as prognostic or predictive factors. For example, extramural vascular invasion. It is defined as intravenous tumor extension beyond the rectal wall, and it is the negative prognostic parameter. Also, for rectal cancer, it is interesting that lymph node size is not sufficient to say if lymph node is pathological or not, because around 70% of them lower than 5 millimeters are pathologic. Here we can see distribution of these parameters in our cohort. We can see that the majority of patients had T3 and positive disease, advanced stadium of disease. In order to compare responders and non-responders, according to them, we found that negative predictive factors for extramural vascular invasion, tumor morphology, when polypoid and semi-annular tumors were compared to annular and stenotic character of the tumor. Also, we tried to combine them and to find some predictive model, and the best one was a combination of tumor morphology and stenotic character, which produced AUC value of 0.70. Next, in order to retrieve more information from images, radiomics was applied. It aims to exploit information on standard imaging that cannot be detected visually. Here we can see the workflow of radiomics. In our case, it started with initial MRI. According to them, some parameters were calculated, and according to them, models were constructed. Our aim was the investigation of the radiomics value in predicting the response of locally advanced rectal cancer, to increase the usefulness of imaging, and the concept of using radiomics lays in the fact that it provides the signature of the tumor. According to the imaging, using radiomics, features are calculated. Feature is information which we get using radiomics. There are two kinds of features, first and second order. First order texture features are statistical measures of pixel intensity distribution in an image, and the example is mean. And on the other hand, second order texture features capture pixel spatial relationships, and homogeneity is the example. In our study, initial pelvic MRI DICOM images were available for 71 out of 75 patients, and 3D T2 weighted contrast sequence were used for analysis. We can see that there were images from different institutions. On one side, it is disadvantage of our study, but at the same time, it's advantage because the approach can be widely applicable. The first step was importing MRI DICOM files into software, 3D slicer, and defining the volume of interest. The volume of interest or region of interest in our case was a primary rectal tumor. According to them, tumor morphology was quantified by calculating a total of 2,092 shape first and second order radiomics features. Here we can see the process of segmentation or defining the volume of interest. On the left side, it is original MRI image, sagittal plane. Here it is the primary rectal tumor. We are defining the tumor on every transversal plane where tumor is visible. And finally, we get this green area. It is the primary tumor after segmentation. According to them, software is calculated features. I said that 2,092 features were calculated. And in order to select features with the best predictive value, lasso was employed, and it produced a model consisting of 8 features, and they are listed here. Here I want to show the visual comparison of one of the calculated features. It is gray level non-uniformity. It quantifies the degree of variation in gray level distribution within the region of interest. It is the first patient where we can see that within the tumor, gray level distribution is evenly distributed, and it represents the low level of this parameter. And it was associated with good response to chemoradiotherapy. And the second patient, we can see that gray level is unevenly distributed. It is the high level of this parameter, and it was associated with non-responders. One more example, complexity. It measures the complexity of the distribution of gray level tone differences within the ROI. Again, the first patient, we can see evenly distribution, low level of parameter, and association with good response. And here we can see more white and more gray levels or zones within the ROI, and it is the high level, and it was associated with non-responders. Finally, models produced by LESO were further narrowed by multivariate regression, and combination of two of them as a result gave an AUC value of 0.76. Also, we tried to combine radiomics and non-radiomics features with features available from a routine radiological report, which produced the AUC value of 0.81. As a conclusion, the application of radiomics in LARC holds potential for assisting clinicians in tailoring treatment plans and making informed decisions for individual patients. As we see that the best AUC value was in combination of clinical or radiomics data. It provides a foundation for future multimodal prediction models, which will also incorporate genomics, proteomics, and metabolomics data. But there will be need for a larger cohorts to validate these preliminary findings. All these analyses in more detail will be presented as a part of training at our project. It will be held at the end of this year. Thank you for your attention.
Thank you so much, Madam. So maybe there are questions to the speaker. Maybe you can, are there any questions from the audience? Perhaps you can unshare your screen, Madam. That may be easier to the other participants. So actually, if there's nobody, no questions for now, I have a question regarding the watch and wait strategy that actually Milena also mentioned. I've heard about this strategy. It's also used to treat, for instance, multiple myeloma, and now recent observations aim at maybe incorporating some kind of early screens to detect like mutations and then incorporate some kind of medication and treatment for the patients even before they actually develop any like detectable signs of the disease progression. Is anything like that also studied in the view of rectal cancer?
Yes.
Screening?
Yes, for this time, the problem in locally advanced rectal cancer is the fact that a clinical complete response not correlates well with pathologic complete response. And at this moment, the only way to say that it is clinical complete response and that there is no residual malignant cells is to follow up patients. There is a protocol for strict follow-up. We are doing control MRI images every 3 months in combination with proctoscopy examination. But we are doing towards combination of imaging and maybe ctDNA level to be sure if there is minimal residual disease or not. But also it is important to say that watch and wait approach, there is a need to aware the patient of the fact that if there will be disease recurrence, there is an indication for surgery. It cannot be omitted. And for some patients, it is for that they will follow up for every 2 to 3 months. It is tough for them. And for this patient, it's better to not doing watch and wait, then perform the surgery and be sure that it is clearly pathologic complete response. But some new diagnostic tests like ctDNA or something else will be beneficial. Maybe
Yeah,
Milena
if I can
add
follow
something.
up as well, Mladen, thank you. So the short answer here for the molecular testing and decision, no, it's still not applied in rectal cancer because we are not quite sure what we would offer as a you know, a specific tell that the cancer has come back. So there's this tumor informed approach where you could track the mutations that were present in the primary tumor and then try to track them afterwards. That's one approach in the ctDNA, and the other one is the tumor agnostic one, just, you know, sequence and find some mutations or changes that are clinically targetable. So it is something that we are working towards in another application for a project. I think this is the future because it will guide us very well. Of course, we will never be able to use only the molecular testing. We will need to combine it with the clinical and imaging, but I think that is the future for rectal cancer and for these patients. At least that's my view. Thanks.
Thank you. Are there any other questions from the audience? Well, okay. If not, then maybe Alexandra will now take over and start her presentation. So
Yes, thank you. I will just try to share it to see if you're able to see. If everything is okay. Can you see my screen now?
Yes, I see your screen.
Okay. The slides are changing?
No.
No. Okay. Here you can see it.
Yes.
Okay. So then we will use the same way. Okay, firstly, I would like to thank the organization committee of Translacore Cost Action. This is really nice opportunity, especially for us, which are early career researchers, to have opportunity to present part of our work from our PhD project. My name is Alexandra Stanojevic, and I'm a fourth-year PhD student from the Institute for Oncology and Radiology of Serbia. I'm working at Department of Experimental Oncology, and today I will try to show you how we used in-depth proteomics approach to profile response to neoadjuvant chemoradiotherapy in locally advanced rectal cancer. So, okay. Let's start with some basics about mass spectrometry. As we already know, this is an analytical tool which measures mass to charge ratio of one or more molecule present in the sample. It can be used for identification, characterization, and quantification of proteins and their post-translational modification. Mass spectrometry can be coupled with different techniques, and especially the separation techniques. One most known is coupled with liquid chromatography, where we are using liquid chromatography to increase depth of analysis, and it is coupled with mass spectrometry, the two MS specters actually, and in that way it is provided to have sensitive high throughput platform which enable analysis of several thousand proteins from individual sample. I really like mass spectrometry because it enables us to see the phenotypic profile of the cell, and nowadays there is so many focus on NGS analysis and genes, and this is really nice and really important, but I really think that in the future years we will go from genome to the proteome very fast. In oncology, there is a couple of techniques which are commonly used. Data-dependent acquisition is the most popular one. We also have one targeted approach, which is single or multiple reaction monitoring, and data-independent acquisition mass spectrometry. Today, I would like to cover similarities and differences between data-independent and data-dependent, as data-independent is next-generation proteomics, let's say, and has some really nice advantages. So in comparison to traditional data-dependent acquisition mass spectrometry, data-independent has really high reproducibility in one injection. One really important point is that with data-independent acquisition, we can do experiments between multiple labs and multiple experiments in one group, and to have high reproducible data. With data-dependent acquisition, this is a disadvantage. The reason is that we are, after the first MS specter, we are using predefined fragmented ions, which are fragmented in the second step. With data independent, as name says, we are using the wide range of isolation windows, and in that way we are selecting all detected precursor ions and we are doing the fragmentation in the next step for all of them. Data independent acquisition mass spectrometry also enable to do the proteome from FFP tissue sample, which was really challenging for us, as we are working in field of oncology, and FFP sample is common type of sample for many diagnostic procedures. So when it comes to science, they are kind of consumed, and we have really low amount of material to work with when we talk about the science. So it was really advantage, because even we had samples which were highly consumed, let's say, we were able to do proteomic analysis. But as any other method, DMS also has some disadvantages and advantages. And the advantages I already mentioned, that reproducibility is at the first place, definitely. But we also have lower number of missing values, because we are using the wide range of MS score for second round of mass spectrometry, and also there is a lower limit of detection. But the disadvantage of this method was really important a little bit before, because at the beginning it was a need for preparing spectral libraries which can be used to identify which peptide correspond to which protein, and before it was necessary to do experiments in your lab to prepare a spectral library and then to perform analysis by your own. In that way, you're increasing cost, you're increasing material consumption, and also we are having like more time to do the experiments. As input for DMS, we can use fresh frozen tissue, FFP tissue, patient-derived explants, cell cultures, and also it can be used for molecular characterization. Sorry, can you see the right part of my screen? Yes, okay, I can. Okay, so it can be used for molecular characterization, biomarkers discovery, therapy response monitoring, and for integration of multi-omics analysis. So what was the aim of our study? I will not go into the clinical details, as Milena and Mladen already explained what is going on the clinical part of our cohort. I will only repeat that we had a sample of patients from locally advanced rectal cancer, and all of them received neoadjuvant chemoradiotherapy. The response to therapy varies among the patient, and we tried to utilize a sensitive proteomic method, data-independent mass spectrometry, to extensively analyze large proteome in terms of predictive potential, also seeking for individuals with favorable initial response suitable for weight and watch approach. Here you can see overview of our methodology. So we have a patient who is going to the diagnostics part, where we obtain the biopsy. So we have a FFP sample from those patients before he starts his therapy. So after that, the patient is going on to chemo and radiotherapy, which is followed by total mesorectal excision. And during the surgery, we have a pathological examination of the response using the Mander tumor regression grade system. So the patients were divided in responders and non-responders depending on tumor regression grade status. So we had tumor regression grade 1 and 2 as responders, and tumor regression grade 3 to 5 as non-responders. Our cohort included, it's not listed here, but included 97 patients, well characterized. And we decided to choose the best candidates from the group of responders and the worst candidates from the group of non-responders, to be able to see the most different proteomic profiles from all of them. As I would highlight that this is the discovery cohort, so it was the first step of our analysis. So the protein isolation, digestion, and SDS page electrophoresis actually purification, was performed in collaboration with our partners from Greece at the Biomedical Foundation Academy of Athens. I was in their lab for a couple of weeks, so we performed all those experiments. We prepared samples for the MS Spectre, and the second part of analysis, MS analysis, was performed in collaboration with one another institute in Greece. This is Alexander Fleming Institute and Professor Martina Samiotzaki, and we performed their LSMS analysis and data processing using data-independent acquisition neural network. This is also one software which enables us to use neural network to identify which peptide corresponds to which protein. This is really deep learning. I will not go into the detail of this really complex software, and it was followed by data analysis, which included Perseus and GraphPad Prism. Also, we use some other softwares publicly available to better characterize our results, but I will show you them. So data processing was also performed in collaboration with our colleagues. So me and my colleague Vasiliki, we performed data analysis here in Belgrade. It was for that purpose we used Perseus Mass Quant software. We decided to do filtering. The first step included filtering of 50% valid values in at least one group of responders and non-responders. The main reason why we choose the 50% instead of maybe higher percentage is that we had FFP tissues, which are, as I said, kind of highly consumed. Let's say we had a material, but still, and also we didn't want to lose some important clinical information. We performed log2 transformation as a standard protocol, and missing values were replaced with imputed values. The modified student t-test, known as the Welch t-test, was used for unequal variance, and we obtained differential expression protein analysis, and protein were classified as a differentially expressed if the p-value were lower than 0.05. There is some question we try to answer. Firstly, how many differentially expressed proteins are in responders and non-responders? And can we use proteomic profile to identify which patient correspond to which category, responder or non-responder? Also, we asked ourselves, is there any maybe drug-targetable proteins, especially in group of non-responders, which can be maybe used off-label to increase probability of better outcome for those patients, and also which signaling pathways are altered in responders and non-responders. This was really important, especially because mechanistic approach behind the therapies is still really not clear. And also we asked ourselves, is there any interactions between proteins within a group apart from signaling pathways? And is there any correlation between protein and gene expression using in silico method? And we also expanded this to wet lab analysis on selected candidates. So our results, we were able to identify 4,300 proteins from 20 rectal cancer FFP samples. I would say here just that we started with the 24, but during analysis we decided to exclude 4 of them due to blood contamination. The samples were not really high quality for mass and LSMS analysis, so we ended up with a 20 samples. And after statistical analysis, we found that there is a 915 differentially expressed proteins between responders and non-responders. From all of them, 700 was overrepresented in group of non-responder, and 250 were overrepresented in group of responders. Here you can see the PCA plot analysis, and you can see that there is a grouping between responders and non-responders. I will only pay attention on these 2 samples. This is the 70 and 67. Initially, we decided to include these 2 samples, but they had the lowest amount of detected proteins. So in a future work, maybe it should be considered to which amount of detected protein should be the best, let's say, cut off for further analysis. As in all results, we found that these 2 samples are kind of outliers, but still they are here. So also on this volcano plot, we can see the differentially expressed protein and the top candidates in both groups marked. And here we also can see the number of identified proteins before and after 50% filtering. So we try to see, can we use the proteomic profile to identify which patient will be responder or non-responder? Firstly, I started with p-value lower than 0.01 and with this protein who met the criteria. And here you can see a kind of clear grouping based on the response. We still have two outliers. This is a sample 67 and 70, and they are classified wrongly. So they are classified as responders. Actually, they are non-responder. But here we try to do hierarchical clustering analysis using the top 10 candidates. And here you can see more clear grouping. We also have one outlier, but this is a different branch. This is not the outlier from the same branch, but still this is the sample which I mentioned is outlier from the beginning. Here we found that proteins listed, actually proteins encoded by listed genes show a high predictive potential of unfavorable and favorable response to treatment. We wanted to see if some of these proteins are already known as a drug target for some other disease or condition. So we performed different analysis to see if they are already targets, and we found a couple of them that are targets for some other conditions apart from cancer, and all of them were or under experimental examination or approved. And still we can see here that all of them show the high statistical significance in terms of predictive potential of bad outcome of the treatment. Next, we decided to do signaling pathway analysis to see which signaling pathways are altered in response to treatment. So we done the analysis on all differentially expressed protein and separately to the responders and non-responders. And this is something also which motivated me to apply for this action, as we saw that there is many signaling pathways related to translational control, metabolism of RNA, and also cell cycle signaling, cell cycle signaling was kind of expected, as we know the mechanism of radiation therapy. And also in non-responder group, we found that it was characterized by signaling pathway related to vesicle-mediated transport, neutrophil degranulation, homeostasis, coagulation, and so on. Also, we were looking for the interaction between proteins. So we wanted to see how they interact between each other. Now you're not able to see clearly because it's not in a PowerPoint slide mode, but let's say like this, okay? So we wanted to see is there any interaction between proteins in group of responder. This was quite interesting, as the responder group was characterized with 200 overexpressed protein compared to non-responder group. But when we performed string analysis, we found that those proteins really interact highly between each other. And here we can see the proteins involved in mRNA processing and genes whose protein products participate in transcription, especially when it comes to pre-MRA processing. Also, factors included in alternative splicing, ribosomal proteins, and factors involved in DNA replication process, as well as factors for organization of proteasomal system. When we do the same analysis in non-responder group, in non-responder group, it was quite interesting. There was not so many interactions. We found a statistical significance that there is a more interaction than it was expected, but still it was not even, it was really different from responder group. Even we had much more differentially expressed proteins. Here on this picture, we excluded the proteins without any interaction to simplify representation. And also, as was kind of expected, we found the protein branch with a central KRAS protein, which is already known to has an important role in tumor progression and development and progression of colorectal cancer. But we also found the protein which are involved in fat and lipid metabolism and components of complement. Next step forward was we wanted to see is there any correlation between protein and gene expression. And when I had my presentation for the first time in front of the audience, someone asked me, why are you going backwards? So you have a protein, you have a confirmed protein expression, and you didn't decide to do further proteomic validation. You go backwards and you're trying to do gene expression validation. It was quite strange, but the reason why we decided to do that, firstly, proteomic studies has really one lack, is that there is no many studies that are validated using the same methodology, especially in a rectal cancer. There is a study performed on different type of samples, data dependent or data independent, but none of them didn't do the validation using the same methodology. They are always using the immunohistochemistry or some other antibody-based methods, which are really good and nice, but we wanted to see, can we maybe do the correlation with gene expression? Why? We have a FFP tissue, which are available in a clinical practice, and we can do real-time PCR analysis really highly precise. So we are able to do to obtain high reproducible results. And we wanted to see, can we maybe use our data to go step backwards to the gene level and to see if there any correlation. As there was so many protein, 950, it was really hard to select a couple of them to try to do correlation. So I used in silico method called in silico software called Rock Plotter. This is the software which combine experimental data from different studies, and I used for patients who have locally advanced rectal cancer and who receive the same treatment as our group of patients receive. And from all of them, I found some candidates which are overexpressed in responder group and non-responder group. There is a top 3 selected with area under the curve, which is quite good, let's say, but still this is only in silico data. We done the same thing with the non-responder group. We also found some candidates to be validated, not validated, correlated with the gene expression profile. And here is the results. So we employed the standard protocol for real-time PCR we are using in the lab on the daily basis. And fortunately or unfortunately, we didn't find a straightforward correlation. There is a preliminary data. The study is still ongoing, but we found that only for one gene, MGLL, we found a statistical trend, but still on the group of 50 patients, it's not as we maybe would say that there is a straightforward correlation. So based on the obtained results, we conclude that there is no correlation between gene and protein expression of the tested genes, which indicate that there is some change downstream of gene expression. We can cause the change on protein level. By the end, without my team, none of this will be possible. So we are the group of people which are really doing together and changing our experience. And I really have opportunity to learn from really experienced scientists. And now we are in the middle of one project, also part of my PhD, where I'm planning to perform meta-analysis of publicly available transcriptomic and the proteomic data and to see what is going on there. So also if you have some ideas or how we can integrate those data between each other or something based on translational control, please feel free to send me an email or my coordinator, and we can discuss about this. Also, this work was supported by multiple projects. And by the end, thank you for your attention.
Thank you, Alexandra. Are there any questions, Alexandra?
Yes.
I have one, if it's okay. So now you showed that the proteomics and transcriptomics, they don't go hand to hand, which is actually nice to see. Yeah, for once. So what would you trust more if you have the choice?
Cool. Maybe I will be a little bit biased, because I really like proteomics, and I truly believe, if I need to say something, I truly believe that we will go from gene level to the protein level, especially because protein are really promising, and I think that it's not explored area. With this advancement in technology, with data-dependent acquisition, I think that all those methodology will go into the clinical practice in the future. So let's say that that I made a decision.
All right. One more short additional question. This is a bit more like technical question. How much material do you actually need for a run, to analysis for proteomics?
So we need a FFP sample, and we need to cut it for 3 or 4 slides of 10 micrometers.
It's not much.
Not too much.
Okay.
Yeah.
Yeah, we were afraid that we would need more samples as well, because for the proteome, of course, you always need more than for the genomics. But in the end, as these methods are now more sensitive, it worked perfectly. For some samples also, you're afraid because we have one FFP only and you want to cut it for diagnostics. You don't want to use it for science. So we wait until everything is done and then you use the remaining material. That's why I agree with Alexandra that this technological advancement that make the techniques more sensitive now are actually helping us with the real clinical samples. Also, for the choosing, of course, proteomics has its value, but we need to also sometimes go back to the genetic level, right? For some things that do not actually go downstream, there will always be the need to perform both studies, I think. So I'm looking forward to the genomic data as well, because we'll do some sequencing and also some fragmentomics to see what's going on. So let's see what happens by next year. It will be exciting.
Thanks. Nice work.
Thank you. Thank you.
Are there any other questions? Hey, thank you for the presentation. It was really interesting, and I really look forward to it. Actually, I'm interested in splicing, and I study that currently, and I've seen that you observe some potential that there might be changes in alternative splicing, so I totally agree that it would be really interesting to look at the RNA level, what is happening there. Yeah. So actually, I do have one question. Just are there any, because you collect the frozen tissue, but is it possible when you have a specimen directly from the surgery to maybe dissociate the tissue and sort a certain type of cells? Are there any surface markers that you could use later, for instance, to follow up and do single cell RNA-seq to check directly for expression of early predictors in a certain subpopulation of the cells? Are there any, I don't know much about the rectal cancer itself, but is it also something that's
Yeah, Alexandra. I'll take this one, Alexandra. I think it's probably better.
Thank
So
you.
we haven't actually progressed towards that, but as we do have a company now that's moving into Serbia for the spatial transcriptomics, we are in route. Alexandra still doesn't know that, but I'm preparing the field for that. So absolutely, we would like to progress towards it. What's an issue here is that actually differentiating these tumor cells from the normal cells within, we don't actually do frozen tissue here yet, or directly from surgery en route. We do it from FFPs. So that introduces a little bit of a technical problem for the downstream application. But if we would to progress, I would really like to see what will happen with this analysis. We don't have it yet. I'm not sure if anybody also from the network can offer that approach. It would be really good. As you know, usually in these countries, that's how we collaborate. The methods that are not available here, we practically apply for other projects or for grants that enable us to get the technique in Serbia. It would be really good. Also, for your question, we would love to hear your thoughts about, of course, the RNA level, as we haven't really touched upon that yet. So if you would be interested in collaborating with us on that, I'm sure Alexandra would be very grateful for that. There's so many things in these omics approaches that you never know what to tackle in which point, because you want to integrate as much data as there is out there, but in the end you will get a workflow of so many omics techniques that what will you produce? What will be the pipeline? What will be clinically relevant? This is something that I'm always thinking about in the end. So of course, all omics are nice, but when is it enough? That's the issue here.
Okay, so are there any other questions in the meantime? If not, then, let me thank you for your participation, for great presentations, and I thank everybody, and see you next time.
Perfect. Thank you, Paulina, for hosting it, and Adolfo as well. It was a real pleasure from our side to be a participant in this webinar.
Thank you
Thank you
very much.
very much.
Thank
Thank
you.
you.
Bye-bye.
Bye.